Well, it seems like in the models that we've been using in cardiac regeneration, as well as others that have been published in the literature, it seems like they play a central role. I mean, the logical pathway for how stem cells work is that they would home to the area of the injury and then differentiate into the cells that were previously lost. But more and more, we're finding that these cells don't last very long. In fact, they release a series of factors and protein mediators that really sort of invigorate the microenvironment to be able to regenerate. And I think exosomes and EVs in general play a central role in that secretome. In fact, in many of our studies, they replace the cells entirely in their therapeutic effect. So there's three central advantages that they have over traditional cell therapy. The first one is their stability. They're not living things, so they don't change by the various stimuli that they are subjected to. And with cells, they change with oxygen concentration, nutrient content, and so on and so forth. And they develop genetic drift over time, whereas these things are just they're a very unique class of biologics. They're virus-sized, and they contain signal, but they're not living things in the sense that we would consider cells to be, so they're stable. The second thing is, the pathways that they affect are quite universal. So the anti-inflammatory and antifibrotic effects that we see in the heart are also seen in other models where fibrosis and inflammation are driving the pathology. Not only do they transcend tissue type, but they also transcend species. So when you inject human exosomes that are therapeutic in an animal model that is non-human, you tend to see the same therapeutic effect. So they're very versatile. And then the final issue is that by virtue of them not being cells, they're not rejected the same way that cells are rejected. So if you were to inject the stem cells that we have, for instance, cardiosphere-derived cells, into a rat from a human, they would be instantaneously rejected. Whereas with exosomes, you can repeat dose without having this whole issue of a graft versus host mechanism that really sort of ruins the entire therapy. There are, I think, 4 challenges. The first one is, how do we identify an exosome out of the many other subpopulations of EVs that exist? You have apoptotic bodies, you have microvesicles, you have other things that are released by the cell in the form of these lipid bilayer particles that may not necessarily be therapeutic. So how do we define an exosome is one key issue. The second issue is, how do we understand the mechanism of action? Because it's a combinatorial effect, these things have classes of RNAs that we don't even know the function of yet. So if the therapeutic effect is a mosaic, we need to understand the individual parts to be able to see the entire picture. We know what the entire picture is, but what are the roles of, let's say, yRNAs or antisense or other classes of newly discovered RNA species? That's a challenge. The third one is, how do we reliably engineer changes in these exosomes to be able to see the effect that we need? That's somewhat hostage to the mechanism of action as well. And the fourth, I think, is a more general lack of understanding of how these exosomes are uptaken in the first place. We know that they're uptaken by all sorts of cells in the microenvironment, but how are they necessarily uptaken by cells from other species? So understanding all of the mechanics that go into EV production, uptake, and signal transduction is going to be key to be able to wield it effectively as we cross from one therapeutic indication to another.